Good afternoon, everyone. A very warm welcome to the webinar series 4 organized by Center for Health Science and Technology at JIS Institute of Advanced Studies and Research, Kolkata. In short, just JIS IASR. Today's speaker in this webinar series is Dr. Oshi Chakravarti, and it is my great pleasure and privilege to introduce her. Dr. Oshi Chakravarti obtained her bachelor's degree in zoology from Benares Hindu University, after which she joined the integrated master's and PhD program at NCBS TIFR. After completing her PhD, she moved to Harvard Medical School for 1 year, and later she joined the NIH. At the Department of Pathology. And after pursuing her postdoctoral research there, she returned to India and became a principal investigator at the Structural Genomics Division at Saha Institute of Nuclear Physics. And there she is continuing as associate professor. Her primary research interest is to understand the molecular pathway that lead to extensive neuronal death in late onset neurodegenerative diseases. She has several peer-reviewed research articles, original reviews, and book chapters to her credit. She is a recipient of the prestigious Janaki Ammal Women Scientist Award from the Department of Biotechnology. Today, her topic is organellar dynamics regulate cellular surveillance. So without any further delay, let us all welcome Dr. Oshi Chakravarti.
Thank you, Dr. Kamakshi, for your kind introduction. So,
yes, I'm just stopping sharing the slide, and now you can share your slides.
Okay, so I will continue with sharing my screen. And since it is mostly students and research scholars who are present, are my slides visible?
Yes.
Okay, so since it's mostly students and research scholars, so I wouldn't mind interruption even during the course of the talk. I will be happy to take questions even if I get interrupted. So because I am most comfortable
sorry to interrupt, but something, Oishee, you are mute now. Could you please unmute yourself?
Am I audible now?
Yes, now you are audible.
Okay, I don't know how I got muted. Anyway, okay. Am I visible and audible?
Slides are not visible, but you
now?
are audible.
Okay, just a sec. Just one sec. Share.
Yeah. So while you are sharing your slide, I just want to add one point. If anyone has any question and want to, they can write in the chat section or ask in between, as Dr. Chakravarti suggested.
Yeah, so if the question gets asked to me by Kamakshi or whoever, then I will definitely answer. Okay. Am I audible now?
Yes, completely.
Okay, great. So initially, I will just introduce you to a few things which most of you know but may not really appreciate. And then I will continue with the intricacies of the interplay between organelles that happens in cells. So the cell organelles, like all of us have seen, the ultrastructure. This was first shown very elegantly. A very high resolution ultrastructure was shown by the celebrated cell biologist Keith Porter, where you could see the nucleus, the endoplasmic reticulum, as you can see, dotted with ribosomes, the rough endoplasmic reticulum, mitochondria, some vesicles, things like that. But if we look at this cell, this is an animal cell, as you all know. So if we look at this cell, things appear kind of stuck in time and space. So these organelles look very distinct in their structure. They don't seem to connect with each other at this level of resolution also. However, as we know, that cells are dynamic entities. Things happen in real time and in real space. So there are changes which can happen at this end of the cell, and they may not be visible at this, the other very distal end of the cell. Similarly, so these local changes affect not just the entire cell per se, but they are actually translated or reflected inside the cell itself. So, to have a better understanding, what we do is we look into the organelles, maybe not in that high resolution. We go a little down on the resolution, but we look in real time. Real time, I mean that as and when the as and when the events are occurring inside the cell. So when we do that, we find that the endoplasmic reticulum, which is green over here, and the mitochondria, which are in red, they are not really discrete. You can see umpteen number of yellow patches. So those are actually interconnections between these organelles. So here I have just given you an example of two organelles, but such crosstalk, such interconnections happen between almost all organelles. So such interconnections, such crosstalks happen between the endoplasmic reticulum and the plasma membrane, between the endoplasmic reticulum and the Golgi, between the mitochondria and the autophagosomes, between the different endosomal vesicles and the Golgi or the endoplasmic reticulum or the lysosome or even the mitochondria. So such interconnections, they are always happening, and not just that, these create microdomains inside the cell, which are actually structurally and functionally little distinct from the general morphology of the rest of the organelle. So, this is where I will leave you with the organellar dynamics at this juncture. And I would like to introduce you to the next thing that was there in the title of the talk, that being cell surveillance. So by cellular surveillance or quality control. So what we know is that primarily there are two very broad cellular surveillance systems. One is the proteasomal degradation system, and the other is the lysosomal degradation system. The lysosomal degradation system is actually getting information from, again, two systems, one being the endosomal system and the other being the autophagosomal system. So usually things which are coming from the outside get degraded at the lysosomal system. Things which are coming from within the cell are getting degraded in the proteasomal system. But this is not written hard and fast in stone, because the autophagosomal system actually regulates things which are happening inside the cell. So these two are basically the surveillance systems of the human, of any cell, of any mammalian cell, or any eukaryotic cell for that matter. So these are quite conserved throughout the species. Now let's go back again to the organelles. So the organelles that we classically know, like and I will take example of just two organelles so as not to complicate this talk, because crosstalks, as I have said, happen between all organelles in the cell. But we will just take the endoplasmic reticulum, and later in the slides I will introduce you to the mitochondria also. So what we know is that the ER is actually the seat where proteins get actually transported inside, and then they get modified and go to various other intracellular locations. It is the place where the calcium storage happens, where lipid biogenesis can happen, where membranes get generated, so many such things. Now, mind you, this is also the place where you can generate more membranes, be it membranes which are required to replenish the plasma membrane, or membranes which are required by the cell to generate autophagosomes, or the organelles where cargo, by which cargo is carried to the lysosome for degradation. The ER is definitely then becomes an important system, because it is actually helping the cell in generating these autophagosomes, which means that it is also important in maintaining cellular homeostasis. So I will actually uh, reiterate on this point a few slides down. Not just that, let me now take you to the mitochondrion. The mitochondrion that we know classically looks like this kidney bean shaped structure or sausage shaped structure under in an electromicrograph. But if you look at the mitochondrion, this is how you can see it's almost like worms wriggling around inside the cell. So this is a real-time image of mitochondria. So what is happening over here is that there is constant fission-fusion dynamics happening at the mitochondrion. And this, mind you, this dynamics happens not only in the mitochondria, it happens at the ER also. And these are actually essential, these fission-fusion dynamics. So where the mitochondria break to form two smaller mitochondria, or they fuse to form a longer mitochondria, and again things can bud off from here. And this fission fusion dynamics is extremely important for maintaining the mitochondrial homeostasis. It is important in maintaining the protein milieu inside the mitochondria. It is important in maintaining its DNA. It is important in maintaining its membrane potential. It is important in maintaining its calcium storage also. Because like the ER, the mitochondria are also the one of the most important calcium storage entities in the cell. So these two are the primary calcium storehouses of a cell. So what we know is that I just told you that fission fusion dynamics happens in mitochondria. So here one was mitochondria of one cell is labeled in green, and mitochondria of another cell is labeled in red. And these cells are fused together, and then you see these patches in yellow where the mitochondria of two cells are mixing. So the fission fusion dynamics of mitochondria happen not just between mitochondria, it happens between the endoplasmic reticulum and mitochondria also. And these are actually seen as junctures which are present between these two organelles. And these junctures are extremely crucial because the calcium buffering of a cell happens through these junctures. If if these don't form properly, then the calcium homeostasis breaks down. Similarly, if too many such structures form, then also it is detrimental for the cell. So the uh, organelle contacts are very important for maintaining the cell health and regular functioning. So what I just said is that the endoplasmic reticulum and the mitochondria, they crosstalk with each other. However, if too much of these cross connections happen between the two, then it ends up in mitophagy. So mitophagy is actually a form of autophagy where the mitochondria are taken inside autophagosomes and they get degraded at the in the lysosomes. So that is what is known as mitophagy. So what I'll show you is that when there is stress inside cell, what we know classically is that if there is stress inside cell, it quite often translates into endoplasmic reticular stress. The endoplasmic reticulum stress is what leads to UPR, as you classically know. UPR, or the unfolded protein response. The unfolded protein response is actually a functional outcome. But at the structural level, what we have just started to appreciate is that the endoplasmic reticulum itself undergoes extreme changes in structure. What happens is that if you look in real time, this is a normal cell. So here, if you follow this white arrow, what you find is that one little portion of this reticulature or one little strand which is coming, it forms contact with the neighboring part over time. However, if the cell is stressed, like if there is a calcium induced stress. What do I mean by calcium induced stress? If calcium from inside the reticulum comes out into the intracellular, into the cytosolic milieu, then what happens is that the cytoplasmic calcium level rises, and that is actually detrimental for many cellular signaling pathways, because calcium being the second messenger, one of the primary second messenger systems, is actually regulating umpteen number of cell signaling pathways. But at the same time, there is gross structural problems in the endoplasmic reticulum itself. So what happens is that, see, if you follow in the lower panel, if you follow this white arrow, over the same period of time, these two don't connect back with each other. So if this keeps continuing, then instead of the nice fish net kind of reticulature, which is actually the hallmark of any endoplasmic reticulum, what you end up seeing is like these large holes in the reticulature. When these large holes form in the reticulature, the endoplasmic reticulum expands. So here you can see like this is like a Y junction, this is classically known as a Y junction, which is formed in the endoplasmic reticulum. But when there is stress, when there is calcium induced stress or other types of stress also, but we will talk primarily about calcium induced stress because that actually percolates to the mitochondria also. So So these junctures, these Y-shaped junctures don't form. And then you get a very vacuolar kind of endoplasmic reticulum. And that endoplasmic reticulum actually expands also till the far boundaries of the plasma membrane. If classically in a normal cell, the endoplasmic reticulum is present in about 70% of the cell surface, when the cell is stressed, it can extend to about 95% of the cell surface. So, which means the surface area, which means that the endoplasmic reticulum, the homeostasis at the endoplasmic reticulum is not functioning properly. And that actually results in another type of autophagy, which is called ER-phagy or reticulophagy, which means autophagy or autophagosomal degradation of the endoplasmic reticulum itself. So it was previously known as ER-phagy. Now it is more commonly called the reticulophagy. So this is what happens when calcium levels inside the cytosol rises. And a similar rise in, so what we know is that endoplasmic reticular stress causes ER vacuolation and ER expansion. Now, the calcium, cytosolic calcium induced stress actually affects mitochondria also, because that is also a calcium storage organelle. So what happens is that when in a normal cell, mitochondria are spread throughout the cell in a high cytosolic calcium concentration, the mitochondria get perinuclear clustered. So how does this perinuclear clustering of mitochondria happen under high cytosolic calcium conditions? There have to be some proteins whose regulation is not happening properly. So we took a few proteins. We went ahead and found that a few proteins were not functioning properly. One of them was a protein which is classically involved in the regulation uh, of removal of bad entities or unfolded, misfolded, wrongly modified proteins from inside the endoplasmic reticulum. So, when there is too much of unfolded proteins present or misfolded proteins present inside the endoplasmic reticulum, these proteins are actually thrown out of the endoplasmic reticulum by a process of degradation, which is known as ERAD or ER associated degradation. So, what happens is that these unfolded, misfolded, wrongly modified proteins string out through channels from the endoplasmic reticulum into the cytosol. At the cytosolic phase of the endoplasmic reticulum, they get ubiquitinated. These ubiquitinated proteins are then degraded by the proteasomal degradation. So, at the endoplasmic reticulum itself, there are umpteen number of membrane proteins which help in this process of ERAD. So one such is an ERAD associated E3 ligase, which is called GP78. The GP78 is actually also present in the plasma membrane, and it is actually associated with some tumors also. But what we found is that the GP78, if there is overexpression of GP78, what you find is a perinuclear clustering of mitochondria like you get when there is very high calcium present in the cytosol. And this is actually, this high GP78 expression can actually be, the mitochondrial clustering phenotype can be replicated by depletion of yet another protein, which is a cytosolic protein called the MGRN or mahogany ring finger protein. So this ring finger protein and this GP78 both are ubiquitin E3 ligases. MGRN is a cytosolic ligase. GP78 is a membrane ligase. Now, so both of these are actually E3 ubiquitin ligases. E3 ubiquitin ligases are present in the cytosol, like MGRN is present in the cytosol. They can be present in the nucleus. They can be present at the endoplasmic reticulum. They are also present at the mitochondria. What we know about these E3 ligases is that one E3 ligase can have multiple substrates. Similarly, one substratum of an E3 ligase or substrate can also have multiple ligases. So, GP78, if there is an overexpression of GP78, you see exactly what we see under when there is high calcium and we see perinuclear clustering. Similarly, if there is depletion of MGRN, we see there is perinuclear clustering. So what happens basically is that and that perinuclear clustering, if you look in the distribution of mitochondria throughout the volume of the cell, again we find that if there is very the mitochondria are clustered, so naturally the volume of mitochondrial distribution will decrease. So when there is high GP78, or if there is the cytosolic E3 ligase go on wrong, both have the perinuclear clustering. Now, what we find is that this E3 ligase actually degrades the GP78 E3 ligase under normal situations, and that happens via the proteasome. And this happens in a calcium sensitive manner. So when there is low calcium, this ligase can degrade this ligase. So when there is low cytosolic calcium, the cytosolic E3 ligase can degrade the endoplasmic reticulum E3 ligase. However, when there is high calcium, then this ligase cannot ubiquitinate and degrade this ligase. And that results in perinuclear clustering. And this entire interaction is actually regulated by a calcium uh, uh, regulated protein, which is called calmodulin. So the calmodulin, what happens is that I will take you directly to this. What happens is that this is calmodulin actually. So calmodulin binds to the cytosolic E3 ligase. When there is high calcium and the calmodulin is present in the hollow form, then the calmodulin binds to the GP78. So calmodulin and GP78 interaction happens. Calmodulin and MGRN interaction happens. Then, sorry, here MGRN and GP78 were interacting under low calcium. So when there is apocam or the low calcium condition, calmodulin is not bound to calcium, then MGRN and GP78 interact, and this interaction results in the degradation of GP78. But when there is excess of calcium, then the interaction between MGRN and GP78 is compromised because calmodulin is masking both their interacting surfaces, and this results in a lot of problems, not just perinuclear clustering. What happens is that the perinuclear clustered mitochondria, they actually go for degradation by the autophagosomal pathway. So what we see over here is that the autophagosome is in green. The autophagosomal protein, the protein which marks the autophagosome is in green, LC3, and mitochondria is in red. So this is a normal cell where you can see just one instance where the green is engulfing the red, but here is a stressed cell where there is either high calcium or low MGRN or high GP78. Then the number of red engulfed by the green have tremendously increased in numbers, which means that there is too much of mitophagy events. So so what we see is that one uh, protein which is actually involved in ER homeostasis or unfolded protein response at the endoplasmic reticulum is actually regulating mitochondrial clearance or mitochondrial autophagy inside a cell. So if you are influencing the endoplasmic reticulum, you can see effects at the mitochondria also. I will further elaborate on this. So here, just to clarify, the MGRN and GP78 interact. GP78 gets degraded under low cytosolic calcium conditions. But if there is high cytosolic calcium, there what happens is that the calmodulin will bind here. The calmodulin will bind this one also. These two cannot see each other. They don't get degraded. When this degradation does not happen, then the mitochondria go for degradation. So why should that be the case? So what we know is that when there is high calcium or when there is high GP78, there are excess of mitochondria contacts. So here in a normal cell, you can see that in green contacts with mitochondria in red, but that becomes practically almost like yellow patch here when there is either high GP78 or there is high calcium. So what happens under such condition? What we have seen is that the mitochondria, as you know, has two membranes. The outer membrane, as we can see over here in red, and the inner membrane, which is in green. So in under normal circumstances, the outer membrane completely engulfs the inner membrane. But if there is high GP78 or there is high calcium, the outer membrane actually becomes unstable. It starts flaking off, and then the naked inner membrane or naked unstable inner membrane gets exposed. Like over here, you can mostly see things in yellow, that there is a outer membrane which is in red and the inside is green. But in these two situations, you can see there are these absolutely exposed, like my pointer is pointing to a green which is absolutely exposed. It does not have any red engulfing it, surrounding it. Again here it is absolutely green. So the red outer membrane is not there. You can find red, small red blobs which are devoid of green, and you can find green which is devoid of red, which just shows that you have actually generated these naked mitoplasts. Mitoplast because it does not have the outer membrane anymore. So when just by regulating this endoplasmic reticulum ligase, E3 ligase, you have actually managed to destabilize mitochondrial outer membranes and mitochondria and generate naked mitoplasts. Just to clarify, what in this ultrastructure, what we see is the endoplasmic reticulum is present here. Mitochondria is here. They can be in close contact in normal cells, but when there is high calcium or high GP, what you find is that the mitochondrion is actually inside another membrane. And here we can see mitochondria are inside the endoplasmic reticular vesicles a vesicle formed by the endoplasmic reticulum. So what we can see in real time is that in a normal cell, these contacts are formed, but they do not get stabilized. What happens is that the endoplasmic reticulum, which is marked in green, actually engulfs a mitochondria, which is in red over here. Rather, I must say this is actually a mitoplast over here, which is getting engulfed by the endoplasmic reticulum. So what is happening, just to show you here, the three colors, what these three colors indicate, the green is the endoplasmic reticulum, the red is the outer membrane of mitochondria, the blue is the inner membrane. So in a control cell, what you have is, so if this is the blue, which is the inner membrane of the mitochondria, you the outer membrane in red is actually surrounding the inner membrane completely, and then comes the endoplasmic reticulum. However, when there is very high GP78 or very high calcium, what you find is that the inner membrane is present, the outer membrane is hardly there, and then comes the inner membrane is now in direct contact with the endoplasmic reticulum. So just to show in super resolution microscopy that you can see, so this is actually a very high, so the images, most of the images shown so far were confocal images. This is a super resolution image, so it does not give structural clarity as of an electron microscope, but it does give better than the confocal. So what you have is that here you can see this is the endoplasmic reticulum. These are small pockets which are formed by the endoplasmic reticulum, and the mitochondrial inner membrane in red is actually sitting inside these pockets. So what we think is happening is, under normal situation, the mitochondrial membrane is intact, and and these two are separate compartments. But when there is stress, calcium stress, then the endoplasmic reticulum actually engulfs this mitoplast, which has its outer membrane broken. And we checked all that with proteomics studies also, and finally to show that yes, the outer membrane is engulfing the outer membrane is not there. The inner mitoplast is actually engulfed by these phagophores, and this is now termed a new pathway, which we have identified as it has now been established, and people have accepted this term as reticular-mitophagy. Why do we say reticular-mitophagy? It is actually two types of autophagy happen together. A part of the endoplasmic reticulum is getting degraded, a part of the mitochondria are getting degraded. So both of these degradations happen simultaneously, and that is why we we call this pathway reticulomitophagy. So organellar contact between two organelles, the endoplasmic reticulum and the mitochondria, is actually helping the turnover of two organelles simultaneously, the ER as well as the mitochondria, through this pathway of reticulomitophagy. Now, stress not only affects endoplasmic reticulum, stress also affects mitochondrial dynamics. So I told you the fission-fusion balance is actually maintained inside the cell. But if there is mild stress inside the cell, it actually results in hyperfusion, or very long thin strands of mitochondria. If there is prolonged stress, then it ends up with fragmented mitochondria. And here you can very well see that here there is a very long, thin mitochondria, which are kind of forming one solid mass. So this is the result of stress inside a cell. Why do I say that? How does that become important? What happens is that I told you that mitochondrial fission fusion keep happening. So in a normal situation, be it the top panel or the middle panel, you can see one mitochondrion breaking into two over time. But during the same time frame, if you look, the breaking of this long strand does not happen. It happens at a much later time. So what you end up is that if you look in a cell, in this cell, you will end up finding these very long, thin strands of mitochondria. And these long, thin strands of mitochondria are formed because the endoplasmic reticulum actually forms a constriction around these hyperfused strands on which a mitochondrial fission protein can sit at the neck and kind of form a tight knot and divide these into two. But that is what is not happening over here. So here the mitochondrial fission protein is marked in green. The mitochondria are marked in the blue over here, and the endoplasmic reticulum is in red. So what you find is that there are far few of these green dots, and hence far less of these green dots sitting on the mitochondria, and far less division of mitochondria happening in these cells. Not just that, they are also not getting, this protein is also getting regulated in a very different manner in these cells which have the long mitochondria. What we find is that the interaction between the fusion protein which and one of its E3 ligases is actually very different between a mutant form of this fusion protein and the wild type, and because of which the fission protein is unable to form proper contact with the endoplasmic reticulum and the mitochondria. So we end up with these long strands of mitochondria. Now, just to show you, just to reiterate, in a normal situation, the mild stress would form a hyperfused mitochondria, but in a stressed cell, when varieties of stress can be there, then mild stress would itself result in fragmented mitochondria, and that is also because of increased mitochondria coupling and increased calcium transfer between these organelles. So the take-home message finally is that ER dynamics, mitochondria dynamics, mitochondria dynamics regulate the turnover of the endoplasmic reticulum. They regulate the turnover of mitochondria. They regulate the turnover of mitochondria simultaneously, and they regulate the mitochondrial dynamics. So the crosstalk or organellar crosstalk or between the endoplasmic reticulum and the mitochondria can have so many can regulate so many surveillance mechanisms inside a cell all together. So fine, I would stop at this, and I would actually like to acknowledge the primary players in this. The talk was based on work of this lady who is now a postdoctoral fellow in Germany, of this lady who is at the NIH now, of this gentleman who is in Switzerland now doing they are all postdoctoral fellows. And this individual who is now almost at the doorstep waiting to submit his thesis. I have not talked about the work of the rest of the people, but that's because of paucity of time. And this work was done in collaboration. A large amount of the collaborative studies were with Dr. Shoika Chakrabarti's lab from CSIR IICB, where we have, because we get a huge amount of in silico help from Dr. Shoika Chakrabarti's laboratory. A lot of these models were generated in their laboratory. The molecular dynamic studies, ab initio modeling was done. So such studies were done in CSIR IICB. A good amount of biophysical studies were done in collaboration with Professor Shomita Basu's laboratory, and the EM studies were done by Dr. Monindra Bera. Shadhin Jana helped us with the STED studies, the super resolution, one of the super resolution images that I showed. Rukmini has been a collaborator since she left. She has helped us with the proteomic studies, and the funding has been from the intramural funding of DAE and the other sources, funding other sources. Thank you very much for your attention. I would love to take your questions.
Thank you so much, Oishee, for such an illuminating talk. You have explained this in such a lucid way. I'm sure that everyone from the students community and everyone has enjoyed the talk a lot. Yeah, so if anyone have any question, please write them in the chat section and we can discuss that. Yeah, so talking about the work, it's really phenomenal. So it's quite new for me, but the explanation was so
yes,
lucid.
the thing is that we think about signaling, we think about functions, but we really do not appreciate the structural changes which are actually very closely coupled with function. So I try to tell my students that when you think about cell cycle, think about the gross structural changes also which are happening at the organellar level, because these are very closely coupled together. Okay, I think there is a question in the chat box.
Yeah. So it is from Shaptoshi Mukherjee. Yes. ER stress causes ER vacuolation. Can you explain the function once again?
Okay, so when stress is there, what is happening is that we really do not know. First of all, I must admit, we don't know why vacuolation is happening. Okay. One of the guesses is that we do not know why vacuolation is happening. The vacuolation is happening because the Y junctions don't form. That we know. Okay. We also know that some of the calcium channels are having a problem. So that we know. But the vacuolation is happening also as actually an epiphenomenon of ER expansion. Why should ER need to expand when there is ER stress? One of the very offhand guesses would be there is too much of unfolded proteins inside. There is too much of misfolded proteins inside. There are too many chaperones which have been upregulated. There are too many other proteins which are required to help refold these proteins which have gotten upregulated. So you have to expand the endoplasmic reticulum. When you start expanding a fishnet also, it has to break in certain points. And that is basically what is happening, what we think is happening during the ER vacuolation.
Yeah, okay. So the next question from Shandip Pal Is the process of reticulum-mitophagy similar in different cell types, such as muscle cells?
Okay, so Shondeep, to answer you, I think we were the first lab to show reticulomitophagy event, because till then people did not uh, uh, see a coupled turnover of endoplasmic reticulum and the mitochondria together. So we showed that in at least three cell types, which was an epithelial cell, a neuronal cell, and an embryonic fibroblast. But coming to your muscle cell question, I am not, I don't know the answer to it, but there are progressively new studies which have come out after ours, which have showed reticulomitophagy to be happening in other cells also. So I don't know about muscle cells as of now, but I think it is a pretty common event that should happen. Actually, coming to the reticulomitophagy event, the generation of mytoplasts, still before our study, mytoplasts had never been shown to actually occur under physiologic conditions. They were only generated in vitro for electrophysiological recording in an artificial system. So this was the first study to show that mytoplasts do form, and now there are other laboratories across the world which are reporting the generation of mytoplasts. So this was something people kind of, I think, overlooked or escaped. So I think reticular mitophagy should be a very common event occurring in many cell types.
Yeah, great. So I was curious about some points. Like, have you seen the role of this reticulomitophagy in tumor progression, or does it lead to cell death also?
Okay, yes, cell death. If there is too much of mitophagy, it will lead to cell death. If there is too much of reticulophagy, it will lead to cell death. So even if reticulomitophagy is not there, even if it's just mitophagy, it will eventually lead to cell death. Okay, so I think cell death, because mitophagy events increase in cell death, and we see that even in our situations also. And now coming to tumor progression, I do not know the answer to it. What I can tell you is that GP78 deregulation is definitely seen in many tumors, in many cancers. So it would be just a matter of time to see that. We are still very new in this, and we don't know, but that is something, it's a good question for an incoming PhD scholar.
Yeah, sure. One more question came. So is there any difference in crosstalk of ER and mitochondria in heteroplasmic cell and homoplasmic cell?
Now please clarify me on heteroplasmic cell first.
So it's for you, Shayantan, please clarify. If you want, you can unmute yourself.
Hello, ma'am.
Yeah.
Actually, by heteroplasmic, I mean that one cell having mitochondria, in which both the mitochondria having a little different genetic material.
Both mitochondria having little different genetic material means mitochondria will have a genetic material, and that will be there in, you mean if two different types of cells are fused together?
Kind of.
Okay, yeah. Then also if there is a proper cell fusion, okay, if you can actually form the cell fusion, like some of the so like in one of the pictures that I showed, then ER crosstalk will ER mitochondria crosstalk will happen, and that should not differ between a homoplasmic and a heteroplasmic cell, because these two organelles, why should these two organelles be differentiating between self from non-self? There is no need reason. Okay. Because
Okay.
if like I showed in the peg fusion assay, if two mitochondria, if two cells are fused together and mitochondria are left to fuse with each other, then eventually, like initially you will see that there is a red fusing with the green, and in the center you will see a yellow zone. But over time, if you leave it, the entire cell will become yellow. So there is a complete mixing of these two mitochondria. And now if you look at the mitochondria junctions also, they will be their genesis will also happen over time. So initially it will be only green fusing with with the of the green cell, red mitochondria fusing with the of the red cell, and then over time it will be yellow mitochondria fusing with the of the entire fusion cell. Now, the material, mitochondrial material inside may be different, but the exchange will happen with the the way it should.
Okay, ma'am. Okay. Thank you, ma'am.
Okay.
Yeah. So I guess no more questions coming, but people are thanking you for such a great presentation. I can see many thank you messages and one more. Oh, okay, one more question popped up. So from
Yes.
Banerjee. So will the fused mitochondria be still functional in ATP synthesis?
Yes, absolutely. We do these we do these studies also, and they will definitely be functioning the way they should, unless they are faulty in some way. If one of the mitochondria, so now you have actually raised a very pertinent question in mitochondrial homeostasis. So what happens is that this fusion fission cycle that continues inside mitochondria at all times is actually very important in actually maintaining the entire ATP synthesis mechanism functional. So if one component of it goes wrong, or maybe two three components have gone wrong, then what happens is that a small portion of one mitochondrion can pinch off and get degraded by mitophagy. The rest of then it the rest of the mitochondria will fuse with some other mitochondrion, and that will actually help in regulating the protein homeostasis, as well as as well as in help in maintaining a functional ATP synthesis mechanism inside mechanism, as well as machinery inside the cell. So this is actually the basis how mitochondria regulates everything inside the cell, everything inside itself rather. It regulates its the quality of DNA, because if there are mutations in its DNA, then it is extremely harmful for the cell itself, because many of these diseases, mitochondrial diseases, are actually associated with mitochondrial mutations. So mitochondria can regulate the DNA by this fission fusion. It can regulate its ATP synthesis machinery by fission fusion. It can regulate its overall potential by fission fusion. It can regulate its entire import export pathway by fission fusion. So all these are regulated by the fission fusion cycle of mitochondria.
Okay, so I guess you just answered the next question that came from Shopturshi. So what is the importance of mitochondrial fusion? Yeah, so I think you just answered this, right?
Yes.
Yeah.
Yeah. And the funny part is so many of you are actually a master student, and some of you are research scholars. What you know is that people talk about probably saying that I have purified the endoplasm, I have purified microsomes. which means that the person is trying to claim that the person has purified endoplasmic reticulum, or the person says I have purified mitochondria, but that is wrong. That is completely wrong. Nobody can purify mitochondria. Nobody can purify endoplasmic reticulum. You can only enrich mitochondria, you can enrich endoplasmic reticulum by fractionation, because you will always get some amount of endoplasmic reticular proteins in mitochondrial fraction, and you will get mitochondrial proteins in endoplasmic reticular fraction always. This is absolute reality. This is truth. We do not have any way of actually purifying these organelles. We can only enrich them. The thing is that uh, under stress, actually many of the mitochondrial proteins get shuttled to the endoplasmic reticulum. Similarly, many of the endoplasmic reticular proteins also can get shuttled to the mitochondrial outer membrane. So there is this crosstalk, this interconnectivity between the two organelles is actually one of the key features which helps cell survive through stress or break down under stress.
Thank you, Oishee. Can I just ask you just a very basic question?
Sure.
So, as we know that mostly the damage associated molecular patterns, I mean the DAM signaling by the mitochondria, which is again a very important thing in case of different brain diseases or something like that.
Correct.
So is there any, I mean, so this calcium kind of upregulation or downregulation, this thing can actually do this kind of damages, and there may be some inflammation. I mean these DAMs can do the trigger the inflammation in brain and might be related to different diseases. I mean,
correct.
The innate immunity and other things will be kind of activated there. So, so could you please, I mean,
so Shondeep, just to give you a kind of a convoluted answer to it, we are looking into one of these things which is very much related to innate immunity, that being the release of mitochondrial DNA into the cytosol or even the extracellular milieu. Okay, so this actually, the extra mitochondrial DNA or it can be extra nuclear also, but we are looking into extra mitochondrial, which is very well known in triggering the C-gasting pathway. Okay, so that is definitely one thing we are looking at, and that is very well coupled with the fission-fusion balance of mitochondria. I mean, I will not go into the details because this is something which is like a story cooking up. It is almost ready to be to come to the market now. So I won't go into the real finer details, but what I can share with you is that if the fission-fusion balance of mitochondria is gone and it tilts towards hyperfusion, then some amount of mitochondrial DNA which can feed into this C-gasting pathway actually gets released into the cytosol. So that is definitely something which is of extreme interest, and that would be very much important in diseases, in brain disorders, mitochondrial diseases, because that would lead to trigger of the innate immunity, that would lead to actually causing inflammation also.
It sounds really fascinating. So we'll listen to it in our later
Yes,
seminars.
you have to wait maybe
we'll wait for it.
longer for that.
Okay. Thank you so much.
Yeah.
Yeah, thank you so much, Oishee, and we are really excited to hear the next story from you. Like activation of the seed-casting pathway by the mitochondrial DNA. It sounds so fascinating. Yeah. So thank you from the entire JIS ISR family, and we are really looking forward for your next talk here.
Thank you very much, Kamakshi. Thank you, Shondeep, and thank you. Thanks to all the faculties at your end.
Thanks so much.
Thank
Yeah,
you.
so with this end, I guess I am ending the meeting here. Thank you.
Thank you. Thank you. Okay.